until it's done. And he said, no, I'll do it right here. Then he said, can you see it? And I said, if I go back to my computer, I can stay there.
Why don't you log in now through Zoom? Don't need to record yet.
All right, for the sake of time, I think we're going to get going. I'll have a few more people I'll be letting in online, and I'm going to try to be monitoring. For those online, the feed that's coming through, so if you have any issues, just type something in the chat box, and I will see if there's anything I need to correct, if I can make the corrections as we go. So it's my pleasure to introduce Leanna. It's Nigon, right?
Yep.
I think I've been mispronouncing that since I know Tyler started, so. So Leanna comes from Wisconsin, did her bachelor's work at UW-Madison, then started with me. I was trying to figure this out. Is it 2015, 16, somewhere in there?
Yep, right at the January 2016.
So then I stuck her in a lab for a few years, and then after that, she finished her MS and then started on his PhD. So I guess for the sake of time, I've seen this presentation. I know you got a lot jammed in here, so
Yep.
I'm gonna let you get started. I do want to thank the Minnesota Corn Research and Promotion Council and Minnesota Soybean Growers. A lot of the work involved here is some of the stuff I was really interested to see the culmination of this, particularly the potassium work. But I do want to thank the sponsors before we start. So with that, I'm going to just turn over to Leanna and we get going with her defense presentation.
Awesome. Thanks, Dan. Like you said, my name is Leanna Leverich Nigon, and I am going to talk to you all about spatial applications for improved nutrient and agronomic management in Minnesota cropping systems. So like Dan alluded to, I have a lot kind of jammed into this presentation. I really had two parts to my PhD project. Part one was about clay mineralogy and soil potassium that I did with Dan Kaiser. And then my part two of my project was on soybean seeding densities, and that was conducted with Seth Naeve. And kind of highlighting broad level across both parts of my research was spatial variability in agriculture. So I'll start broadly by introducing us there. So spatial variability, here's a few examples that you can see on the screen here. Here we have geology up in this top corner, which is more of a coarser resolution, more county level variability. Then we see spatial variability as you can find on the farm fields, such as the soil test potassium level or a digital elevation model known as a DEM, where we see variability over space. And essentially what this tells us is that we're going to see that variability reflected in our yields, like we do here in this soybean field with yield, where we see over space, there's quite a bit of variability happening. And really, spatial variability in agriculture is just inherent. We see it across all farm fields. And we can either block for it with traditional experimental design methods by randomizing, replicating, and then blocking to remove kind of those random effects to really get at a response variable. But then there's also the approach where we can try to capture some of that spatial variability or the features causing that variability and embrace it to look at how it's influencing our response. So there's benefits to really having both approaches. We need that fundamental science to learn about what's happening with our specific response variables, such as soil test levels, but then it's good to also look at the broader agricultural context with the variability. So in both of my chapters, you'll see that I work with spatial data and on-farm data, and on-farm data inherently is spatial. And there are some factors that I pull out that I use to help to explain that variability that's occurring either across the landscape or on the farm. You'll see elevation and DEMs, routine soil test measurements, remotely sensed data, including elevation as well as some imagery, soil and climate layers, management data from the growers themselves, and historical data from the grower. So that would be yield or historical yield maps. So these really kind of underpin all of the work that kind of culminated for each of my two parts. So diving into part one, soil potassium and clay mineralogy. Again, this was with Dan. And we looked at everything from the lab level all the way up to the field level with on-farm trials, and then of course regionally as we tried to map clay mineralogy. So before I dive too deeply into potassium, it's good to just reorient ourselves and remember what are the major pools of potassium in the soil. So we have our soil solution K, which is readily available for plant uptake. Then we have exchangeable K, which is K that's available from the cation exchange complex. It's bound to those clay minerals, those negative sites on organic matter and clays. And that's readily desorbing and adsorbing with the soil solution. Then we have our fixed potassium, which is due to K that's being caught in inner layers in the non-exchangeable fraction for clays, the phyllosilicates. And then our mineral fraction, which is the majority of our K in soils. More than 95% of our soils are in that solid mineral form that is only slowly available through release over long periods of time. So Dan and I kind of identified 4 major buckets from which we can measure potassium, or how these soil features are impacting potassium, and they were clay mineralogy, CEC, and texture, which also captures the percentage of clay in a soil. And they kind of describe this soil holding capacity for potassium. And then we also looked at how other cations are interacting in the soil. How are they moving around and competing for sites on the cation exchange complex? So that brings me to chapter 1, where we look specifically at K retention in sandy soils. So why sandy soils, you ask? There's been a lot of anecdotal evidence that we've seen in Minnesota that shows that our sandy soils or our coarse textured soils are behaving differently than our finer texture soils with more silt or clay. One example of this was published in Proceedings with Jeff Vetch and Dan Kaiser, where they looked at the soils at Becker, and they did a trial where they measured, what is the annual potassium rate that needs to be applied. And essentially here, the recommended rate would have been 108 kilograms per hectare, which is more of this 120 rate that we see in the table, in order to maximize yield. However, yield was actually maximized at this lower rate at 60 pounds per acre, or 56 kilograms per hectare. So this is surprising. Why is it that the sandy soils just did not require that additional potassium? Now, other states like Iowa, Antonio Mallarino's lab have seen similar differences as well with their coarse textured soils. So we decided to specifically look at the CEC, the texture, which plays very hand in hand with the soil CEC, and then other cations that might be influencing potassium retention and availability in those soils. I had 2 objectives in chapter 1, but I'm going to go over the first objective, determining the influence of pH level on CEC and potassium absorption in coarse textured soils. So the methods behind this, we collected 4 soils across the state of Minnesota in the sandier areas of the state. The first soil was Becker, which is plotted here as number 1. It is a loamy sand, and it has a CEC around 6. Our second soil was Big Stone. It has a slightly greater abundance of clay with a CEC of 8.4. Cambridge is also a loamy sand down here, and it is a CEC of 4.5. And Lamberton is a clay loam that is kind of acting like a check. It's very different texture-wise compared to the other sandy textures that we've evaluated. So they were all sampled from low pH areas in the field. And then we limed those soils to create a medium class for pH and also a high class for pH. So we had low, medium, and high for all 4 of those sites. And then we ran sorption tests on each of those different pH levels. The sorption test had 16 sorption levels, 0 to 300 ppm or milligrams per kilogram. And in that potassium solution, we had the background being deionized water or calcium chloride. So what were the results of this? We did see in the calcium chloride that there was an impact of the pH level at every site, really, though the response was a little bit mixed. And then when we look at the deionized water, there was a response, but only at one site. And while pH level was somewhat interesting, what we actually found to be really the driving force was that background solution of deionized water or calcium chloride. So here's a look at our sorption curves. I'll orient you here. We have potassium adsorption or desorption. Of course, when you are above 0, you're having absorption occur. And when you are below 0, you're having desorption occur. And this on the x-axis here is our equilibrium potassium concentration, which is the potassium in solution. So for each of our 4 sites here, we can see that deionized water resulted in something completely different than what we saw for the calcium chloride. So just to kind of walk through this step by step, we first look at our loamy sands. We see overall with the deionized water, there wasn't a whole lot of sorption occurring. There was a little, but it wasn't increasing as we increased our potassium concentration. And of course, with that calcium chloride, those red dots here, we see quite a bit of desorption occurring. Now, the sandy soils, they couldn't sorb additional K, but when we look at a sandy loam, which on the textural triangle, we see that it's a little higher up with the clay abundance, and also it had a slightly higher CEC, an 8 versus a 4 and a 5. That did increase the amount of K retention that we saw across this potassium concentration for the deionized water. If we consider just this divalent cation, calcium, basically it is outcompeting potassium on these exchange sites, or it appears to be doing so, and it is just knocking potassium into solution. Even at Lamberton, a clay soil, we see less sorption occurring for the calcium chloride in comparison to the deionized water background solution. So one last thing to point out, though, is that Lamberton, this clay soil, was able to sorb under both background solutions. It was just able to sorb more potassium with the deionized water without calcium present. So our high level conclusions from this are really that soils with a higher CEC are able to sorb more potassium. When you have a higher clay percentage or a texture that's clay based, you're able to sorb potassium even with a competing cation present. The sands did not have a whole lot of sorption capacity in general, and that these divalent cations such as calcium or magnesium, they will compete with K for sites on the exchange, which is something that, you know, we inherently think of with the lyotropic series, but we don't always see in practice, and this was a good example of it. Something that we didn't directly measure, but we suspected was occurring, is that the clay mineralogy was also influencing our potassium in solution. We kind of suspect that the 2-to-1 non-exchangeable fraction, so that space between the inner layers of clay, was actually catching some of that potassium. So to look at that a little bit more in depth, that leads me to chapter 2, where we looked at refining our potassium recommendations and fertilizer guidelines for corn using soil mineralogy. So in this chapter, I'm going to review objectives 2 and 3, where we determine the impact of smectite and illite clay minerals on the critical K concentrations, and we evaluated the corn grain yield response to K fertilizer, considering the underlying clay mineralogy. So again, back to those pools, we are considering especially this exchangeable fraction, the fixed K fraction. This is very important for our phyllosilicates. And then, of course, our mineral K is always relevant when we're talking about mineralogy. There were really 3 clay minerals that I looked at. These are all phyllosilicate minerals, meaning that they are a layered mineral with silicate. And first we have our kaolinite mineral, which is a 1-to-1, meaning it has 1 octahedral layer and 1 tetrahedral layer. And these stack on one another, and they don't really allow for an interlayer space. They have a relatively low CEC, though this is due to isomorphic substitution more so than this interlayer. Micas and illites are our second class of minerals that we looked at. They have a 2-to-1 mineral layer, meaning that there is 2 tetrahedral sheets on either side of an octahedral sheet. And when these minerals stack and layer, they create this nice space right here. You can see this yellowish ion that would be a potassium ion that fits just perfectly into those tetrahedral holes on the layer. And then the charge is balanced just perfectly to capture that 1+ charge on the potassium. So this is why we call these a fixing clay. They tend to fix potassium. And then our smectites, these are usually considered kind of shrink and swell. They can contain quite a bit of water in their interlayers, thus they have hydrided cations that can flow in and out. And again, they have those 2-to-1 layers. Now these have the highest CEC because they can sorb kind of in multiple areas in the interlayer and also on the edges with a lot of isomorphic substitution occurring. So for all 3 of these minerals, of course, they can weather over long periods of time, and they all have a CEC, a cation exchange capacity. They have negative sites that are interacting with this exchangeable K, but it's really our micas and our illites and our smectites. I'll refer to mica as illite for the remainder here. But they are really the ones that are playing the role with the fixed potassium, these 2-to-1 minerals that can react pretty differently. So in order to look at our smectites and our illites across the state and also kaolinite, we ran 20 K-response trials in 2019 to 2021. And the important part of these trials, I have an example of on-farm trial here, is that there was a limiting rate, which would be like a 0 rate of potassium, and a non-limiting rate, which is our highest potassium rates in order to capture response. So we use those 20 response trials in addition to 23 historical trials that have already been run prior to this experiment for a total of 43 locations where we had K responses at different sites across the state. So you can see they were spread out all over Minnesota. At each of those locations plus additional locations, we took samples to test for the actual mineralogy makeup. Now, this was done using an XRD or an X-ray diffraction analysis in order to determine the relative abundances of each clay mineral, smectite, illite, kaolinite. And this method we use to then determine the smectite and illite ratio at each of the locations across the state. And this ratio, we did a cluster analysis on with K-means to kind of break apart, are there differences in the mineralogy across the state. And we found a smectite to illite ratio of 2.8 is kind of where the data set broke. Now in North Dakota, they did something similar and their breakpoint was 3.5. So we can see that we're kind of in a similar ballpark to one another as far as that breakpoint in the smectite to illite ratio. So then we could classify our field locations as either high smectite soils with a ratio above 2.8 or as higher illite soils, which would have a ratio below 2.8. So once we are able to separate out our data set, we end up with 2 data sets for looking at response to potassium fertilizer. We have our high smectite data set and our high illite data set. And here is our response to potassium soil test potassium level. So on the bottom axis here, we have our air-dried soil test potassium measurement, and we have our relative corn yield on the y-axis. And what we can first see in the high illite soils is the point at which yield stops increasing here is right around 86 parts per million potassium or milligrams per kilogram, versus in the smectite soils, that number is closer to 187 milligrams per kilogram K. So there's quite a difference in where we see the data set just break for a critical point. Something to mention right away is that we see a much greater spread in the smectite data, whether that is inherently because of the smectites or just because it's the data itself is hard to know, but there is more variability in this data set. But essentially what this tells us is that the building max point, the point that you would want to build your soil potassium levels, is about 100 parts per million or milligrams per kilogram less in the illites than it is in the smectites, which if we remember back to chapter 1, those sandy soils, they tend to be more illitic, they tend to have more illite dominated, again, kind of related to this lower requirement. So what we did is we looked back at our critical levels for each field site, and we found that if you use the correct critical level for each field trial, we are able to predict if the site will respond to K fertilization 85% of the time correctly. So what does this mean for our growers? When you have a higher smectite soil, you have a higher critical soil test level for K. Your higher smectite soil may mean that more K fertilizer is required in order to meet your optimum yield goals. In high illites, a sufficiency approach might be more appropriate since we are not able to, or maybe it's not necessary to build those soils quite as high for those illites. Now, knowing that there is a segregation in the data set between smectite and illite leads to the question of if growers should be measuring their clay mineralogy. Now, we suspected that clay mineralogy is not going to vary within field too dramatically because it will be mostly dominated by soil forming factors and geology, the underlying parent materials of the soil. So first of all, we don't expect it to vary too significantly within a field, but also it's not particularly affordable for a grower to go out and sample their fields for clay mineralogy because it is roughly $300 per sample to measure it. So that led us to our next chapter, which was developing a workflow for making clay mineralogy maps using publicly available spatial data and supervised regression. So in this chapter, I will talk about how we developed and trained supervised regression models to predict 3 relative clay mineral abundances, kaolinite, smectite, and illite, and how we use these trained regression models to then predict clay minerals to create maps of the state of Minnesota. So our training data set, you saw this before, but it was classified as smectite and illite high or low, but there was 157 samples. And again, this was an XRD analysis that was run by ACT Labs in Canada. And these minerals reported by ACT that we decided to predict were smectite, illite, and kaolinite. How we did this, we used 24 initial spatial layers to describe the 5 different soil forming factors. So we captured factors that would represent parent material, topography, biota, climate, and time. And all of these factors were collected from public sources, public data sources such as the USGS, the Minnesota Geological Survey, or the Minnesota Natural Resources Atlas. To give you an example of what some of these layers looked like before some initial processing. So this is an example of polygon data here with the parent material lithology. And we can see this is polygon data. And if we look at some of these other rasters that we have here, this is raster data, so it's gridded data. So initially we see that our features that we're going to use to predict are not matching. So we have to do something about that. We create a skeleton raster. You can do this in QGIS, you can do it in multiple softwares, but really what we're doing is we're creating a uniform resolution and an orientation across space for us to summarize our layers on. So what we did is we took all of our raster data and we resampled it down to match our 1 kilometer resolution and also orientation on the world, essentially. And then we also rasterized any polygons. So like that lithology data, instead of being a polygon shape, we turn it into a grid shape, just like our raster skeleton. So once all the features are all summarized down to that raster skeleton, the same distribution across space and size, we have a raster stack, and this raster stack has 24 bands. Each band is representing each feature. And then we lay our sampling points, our 157 points on top of that raster, and we sample each of those bands to create a feature matrix. So all of our 24 features are going across in columns in a new data set or in a data frame, and 157 observations make up our rows. And then of course we have a target vector, which is our smectite, our illite, and our kaolinite, which is that, you know, those would be 3 separate columns as well with 157 observations. So now that we have our feature matrix set up, we're ready to actually identify the features we should use. So what we did is we tried to identify our top features using LASSO, and LASSO is just a machine learning model. It's a regularization model, but it stands for least absolute shrinkage and selection operator. And the goal of a feature selection is that we want to reduce the complexity. So we don't want to include too many features, but we also want to reduce the error. So here we have the number of features on our x-axis, and on our y-axis, we have our root mean square error. So we're trying to see how many features we need to add to pull this error down, but reduce the complexity. So for illite here, we see that as we added 4 features that were numerical, hence they are triangles, we reduced that error, and then adding any additional features really didn't benefit the model anymore. So we settled on our 4 numeric character or features, and then we did the same process for our categorical. So then we added categorical variables one by one until we completely lowered the error as much as possible until it leveled out. And that's how we conducted our feature selection, was just reducing that error. So for illite, that resulted in 8 features. For kaolinite, that resulted in 6 features. And for smectite, it resulted in 8 features to be included in the model. I'm not going to go into the specifics of which features, but I'd be happy to talk about it later. The next step was to run our cross-validation method for training and tuning with only our top features. So with our data frame, we've used a leave one out analysis. And so many of us have heard about, you have your training set and your test set. You hold back the test set so that you can see how your model does with the training data. In a leave one out, we repeat that process for as many samples as we have. So we run all the training and the tuning with 156 samples, and our leave one out sample is our test set. And so that results in having a test set of actually 157 observations, because we've run the model and trained and tuned the model 157 times on each of those individual samples. This is not for every learning problem, but it was a good approach for our data because we did have a limited data set. So looking at our model accuracy, how it performed, for illite, we had an error of about 17%. You can see this model underfit from this 1-to-1 line. I'm sorry, I'll back up. So we have our predicted values on the Y and our observed values on the X. And this is not a best fit line. This is actually just showing how well the model predicted. So this is the 1-to-1 line. So we had a 17% error rate with the illite. We had about a 20% error rate with the smectite, and with our kaolinite, we had about a 5.5% error. We can see where the model struggled here. For smectite, it was these low smectite values that were really over predicted. They were supposed to be 0, but they ranged, quite wide, which brought up that error quite a bit. In kaolinite, we see it over here with our higher kaolinite values that are being observed but not predicted as such. And with illite, kind of the same. Where we have higher illite is really where the model struggled to predict that higher amount. So there's a few reasons why I think that this occurred, that we did have some error. The first thing to point out is that these clay abundances are relative abundances. This is a semi-quantitative method to measure mineralogy. Typically in the labs and the papers that I've read, they say if this is done perfectly, you're going to get a 10% error rate, or 10% error on either side of that abundance that's reported. And that's simply because the X-ray diffraction process is simply a reflection of the X-ray that is then interpreted from using standards. And so there is room for error in there. Using smectite, for example, 10% could possibly be due to the lab, 10% could be due to our modeling. Smectite was on a scale of 0 to 100. So that could be part of it. Another part of it is if we just look at how our data was distributed, essentially. If we look at the relationship between smectite and illite, we see this really nice linear relationship until you get to these 0 smectite numbers, and they are just all over the place on the relative illite scale. Similar with kaolinite and illite, if we look at it, there's this nice solid linear relationship, but then we see this kind of inverse relationship that is, I can't really explain, between 25% and 0%. It's hard to say if that's actually error or if it's really true that these samples are what they are being reported as, but there wasn't really a good logical reason to remove them from the modeling effort. Something that could be tried is you could try to build a model just to look at those smectite values instead, and then build models from 25 and up for looking at those soil samples in particular. So what did this look like, the finished results? So we take these models and we predict each raster cell for the state of Minnesota. And we see that there are, the geographic features of Minnesota are pretty clear. We can see the Red River Valley with the smectite. And the points that are overlaid on these rasters, those are the observed points. Of course, the rasters are all predicted. And we can see that it pretty much follows the trends pretty well. There are a few stickler points here and there, but we see that it's generally capturing the variability across Minnesota. I will mention that illite, most of those values fall around 50 or below, and so it's hard to see some of the differences that are actually occurring in that lower part of the state down here. So same with kaolinite, they tend to be lower values in general. If we zoom that scale into 30 to 0, we might see more of that difference. So in conclusion, with the clay mineralogy maps, the prediction model really worked best for our smectite mineral, which is very promising for potentially using in the future. And we did use the simplistic regularization model, LASO, and that was used and was pretty successful in working to make predictions rather than a complex model. So part 1, overall, we found that there is a need to improve our potassium recommendations, especially with sandy soils. There's room for improvement. We see that there are a lot of features that are impacting that. Our clay abundances, our illite and smectites. So there is room for improvement there. The presence of other cations such as lime, cations that could be in lime or irrigation water, they can impact potassium availability, especially in those sands that we see. So something that we need to consider as we're applying anything that might compete with K, it could move it into some solution. And that, of course, K recommendations in corn, we can use illite and smectite and clay mineralogy to improve our understanding of K behavior. So that wraps me up for part 1, and now I'm going to dive into part 2, which was optimizing soybean seeding densities. And this was done with Seth Naeve in the soybean lab. So the motivation really behind this is that variable technology has been released and it's being used really all over the state. And farmers are now asking, okay, I need to manage within my field because I have the capability to manage within field. And with these precision planters, people are asking, how do we use variable rate technology for seeding density? So that brought us to our first question, which is how do we quantify the spatial response and the economic influence of soybean seeding densities? And in this chapter, I'm going to go over objective 1, which was to determine the extent to which optimal seeding densities varied within individual farm fields. So in chapter 4, and in chapter 5 for the soybean study, we used on-farm trials. They were conducted for 2 years. At 2 sites, we had 2 years of data, and at 1 site, we had 1 year of data. So that was 5 site years total. And these were traditional strip trials that were done with just the farmer's equipment. So each pass of the planter was 1 strip that you can see here. And these were modified to fit each grower's equipment. And then the total area of each experiment was roughly 10 to 30 hectares, which is essentially 20 to 60 acres. So these seeding rates varied from 185 to 432, but they varied between different farms and were kind of tailored to each grower. And then at each site after the experiment, we created these zones by using transects across these traditional strip trial. And that allowed us to look at, okay, what is the spatial variability within each specific zone? And these zones were about half a hectare, 0.4 hectares, for us to look at just those in particular spots in the field. So right here, we're looking at site 1, year 2, and over here on this left side is the yield. The golder colors are the high yields and the dark colors are lower yielding. And essentially what we see is that it's very highly variable. There is a lot of yield variability occurring across the field. And then if we look at, okay, what's an optimal seeding density, what yielded the highest? We can identify, the higher seeding densities in some areas and lower seeding densities, the darker colors in other areas, but they don't particularly match one another. They could be different. And so this brings us also the question of, okay, is this the optimal seeding density, or are there multiple optimal seeding densities? In some of these blocks, it might say that this is the highest yielding, 432, but in reality, there were many seeding densities that were optimal in that block. They all yielded the same. So what we did is within each zone, we conducted a Tukey's HSD test in order to determine which were the seeding densities that yielded the highest. And so anything that was in this A group was considered a high yielding seeding density, or an HYSD. And there could be 1, 2, 3, 4, or all 5. Obviously, if there's one seeding rate, it's a very responsive area of the field. If there are 5 seeding rates in that A group, it didn't respond to seeding density at all. So let me walk you through this. This can be a little confusing, but here we have our number of high yielding seeding densities on the x-axis here. And essentially, if you have 1, it's more responsive. And if you have 5, it's non-responsive. And this is the proportion of the field that had however many high yielding seeding densities. So I have each line here is our site year, and then we have our average across all years. So for example, if we look at this purple line, this was site West 19, about 30% of the field had 1 optimal seeding density. But if we look at, okay, if we count 1 and 2 optimal seeding densities, it's roughly 60% of the field that was towards the more responsive side. So overall, what we found is that in most cases, there's not a single optimal seeding density in a particular zone. There is usually many seeding densities. And to make recommendations, we really have to predict that responsiveness. So that leads me right into chapter 5, where we predicted prediction of soybean yield response to seeding density. So I'll take you back to this graph where we're looking at the responsiveness. And this is what we're trying to capture. We're trying to figure out, okay, where are the areas of the field where we have 1 or 2 high yielding seeding densities, and where are the areas of the field where there's 3, 4, or 5? Can we predict this responsiveness? So I'm going to hop just to objective 3 to cover. And this objective was to train and evaluate the accuracy of using field factors to predict the response to seeding rate using machine learning methods. So what we decided to do was to create a response classification problem, meaning there are with only 2 classes, responsive and non-responsive. And kind of how we decided this is let's look at where the average is across all these sites, and where is 50% of the field crossing, which it was kind of logical. 1 and 2 high yielding seeding densities, meaning that there were only 2 seeding densities that would optimize yield. That was roughly 50% of the field, or the zones that we had. And then the remainder were 3, 4, or 5. And so we wanted those 2 classes. We wanted it to be consistent across all of our site years. And we wanted to have enough data in both our responsive and non-responsive class that we would actually be able to train the model. There were only 207 zones total across all 5 site years. So we needed to make sure that the model would have enough data to even do any type of prediction. So we used very similar features, or some similar features to what we saw in chapter 3. We used elevation and elevation derivatives. We used soil test measures such as pH, organic matter, routine soil test measures. We used some Sergo properties such as soil type. And then we used the historical data from the farmer, historical yield. And so that was their data from 7 to 9 years summarized as the mean yield across space, or the standard deviation of the yield across those 7 years, or the coefficient of variation. And then we used a random forest model to create this, this plot here, which indicates categorical feature, or I'm sorry, the feature importance as you, as this number grows. This is the mean decrease accuracy. And that essentially is saying, as you move this feature around in the model, how much is it impacting your accuracy measurement? That is the error report. So we can see up close here what were kind of these top features. Soil series from Sargo and complex name were both up there in the top 5. Primarily why I think these are up there is because it was differentiating the site years from one another. It wasn't necessarily finding in-field variability. It might have been splitting a field in half, but more than likely it was just differentiating our sites across the state. And then we had our yield mean and our yield CV, which were pretty important. And primarily, you know, in the literature, a lot of other researchers are looking at historical yield and finding that it is pretty indicative of soybean response to seeding density. And then we had a couple of other features that were also important in the model. So with the classification problem, we can create a prediction confusion matrix. And I know this is a lot to look at, but here we have our predicted classes on this side. So this is what the model thought the measurement should be. And here is what it actually was in reality. So 16 of our test set, which was 51, so 51 samples in this confusion matrix. 16 of them were responsive and they were classified correctly. And another 22 of these were classified correctly as true negatives. And so the overall accuracy of this classification model here was about 74.5%. When you only have 2 classes in a classification model, this means that it's only really explaining 49% more than what you might have with random variability. So there's room for improvement in this model. However, something to pull out of this very busy confusion matrix is if we just look at our non-responsive sites. The prediction accuracy for non-responsive areas of the field was closer to 81.5, 82%. And this is relevant because if we can predict the non-responsive areas of the field, then we know where we can apply that low seeding density, where you can save money on your seed cost and you don't get a yield penalty at all. So in general, in chapter 5, we found that we could predict the responsiveness to seeding density about 75% of the time, or with 75% accuracy, and that our historical yield features were important features for response. And overall, in part 2, when we're looking at optimizing seeding density, we find that across any particular area or zone of the field, there's usually not an optimal seeding density. There's usually more than one. Predicting which areas of the field are responsive seems like a good potential pathway forward. Historical yield is important in those model measurements and in characterizing that seeding density response. And also, while this model has room for improvement, this is a workflow that could be replicated with a much larger data set with more than 207 samples that we did have for also predicting response. And you could use this to predict response to anything, not necessarily just seeding density. So in kind of conclusion for this whole dissertation, you know, my studies kind of led me to, you know, trying to use supervised regression models to capture what's happening with the spatial variability across the state or across a farm field. And so with agronomic data, spatial variability is unavoidable. And so it's kind of a natural fit into those types of experiments or types of models. And so I think it's a really great opportunity for us to use on-farm data and embrace the variability of soils, topography, climate, et cetera, to begin to try to capture more of that on-farm response. And with that, I'd just like to thank a few people. My advisor, Dan Kaiser, who has been with me since, like he said, 2015, teaching me how to do research, trying to teach me not to be biased and how to recognize my biases and to keep plowing forward and working ahead even when something goes wrong, which many things went wrong, but a lot of things went right too. Also to Seth Naeve, my co-advisor, who really pushed me through my PhD and helped me to really get a start there. My committee members, Joe, Nick, Fabian, who have supported me. The soil fertility research team, Thor, Darby, Nick, Hatley, you guys have all been a huge help. The SWAC department, Carrie, Carl, Melissa and Andy, they've all been there to support me. Anna Cates, Hava, Blair, and Mehmet, they all provided some soil samples for the mineralogy mapping. The LAAS grad students and APS grad students, you guys are great. of course, my family and friends, my dad, who taught me a lot about farming and about agriculture, my mom, my brothers and my sisters, the Nigon family, my in-laws, Sharon, Nathan Hecht, in here, Erica Ellison, Megan Lawrence, and Jamie Palmer. These are all people who have been really, really supportive to me. And of course, my husband, Tyler Nigon, who also went through this program a few years ago and has listened to every dumb idea I've had and every good idea I've had and still supported me through it all. And then of course, our research and promotion councils, Minnesota Corn and Minnesota Soybean, they do such great work for the growers of this state. And I really appreciate their investment in this work. And Bear, who helped to fund the first 3 years of my PhD. And I would be remiss if I did not mention somebody who's passed away and long gone, my grandpa, who, he really kind of instilled in me this interest in agriculture in the first place. And he had this quote on his wall: "The land belongs to the people, a little to the dead, some to the living, but most of it belongs to those yet to be born." And I think that just kind of drives a lot of my initiative and a lot of my interest in this field and in trying to make agriculture a better space. So with that, I will thank you all for your attention, and I will take any questions that you have. Carl. Thank you.
Very interesting. A couple of questions. On your seeding density, what do you think's going on with calcium? Because you had calcium as one of the.
I think that calcium might be related to general fertility overall. There were, in the times that I run the model, you would sometimes just end up with one soil test measure that was up in the importance. So I think it was kind of kept picking up on general fertility across the field, and it kind of shifted in and out. There was potassium and phosphorus sometimes made appearances there as well.
Yeah, I would, maybe cation exchange would come to the top, but maybe.
Yeah. And I think it's just generally related to that overall fertility.
And then going back to your sandy soil potassium, you'd think that the response on those sandy soils is really due to the type of clay or just lack of clay?
I think it's due to the lack of clay overall, though I do think in the Big Stone and Lamberton that did have clay fractions, I think Lamberton, it could be having some of that, non-exchangeable K kind of interact.
That was a clay loam, wasn't it? Or was that a sandy soil?
No, the Lamberton was a clay loam.
Yeah, that was clay.
Yeah.
But I'm just thinking within the sand, it seems like maybe more lack of clay. I mean, did you have sands that had different illite to smectite ratios?
For the most part, they all kind of fall into that
Same,
lower, like more illite dominated class. So there wasn't anything with like high clay, so it wasn't particularly surprising that it did not sorb a ton.
Right. So I guess it's just mainly lack of clay rather than type on the sands.
Yes. In the sands. However, I would say maybe what you're getting at is this illite smectite ratio.
Yeah.
Basically, what illite is characterizing is sand. It tends to be, there are illites that are higher in not sandy soils, but the illite dominated soil was typically in the areas of the state that would have been, our central sands. So they kind of just fell in similar classes.
Could you just do rather than looking at the type of clay, could you just do it on the amount of clay in a soil?
So that's kind of the next question is do we really need to do the mineralogy analysis, or is it related enough to just general clay abundance? I think by running all this, we found that, okay, well, smectite is highly related to your clay abundance. So maybe that's enough to just get a head start, is just identifying your texture overall before you even move into looking at the specific types of clay. Yeah.
So you also measured K-bearing feldspars with this too, but you didn't show any of that data up there. Can you just talk a little bit about why?
Yeah, so we did measure feldspars, but they were part of so there was two different measurements reported by ACT labs. There was the clay fraction, which is what I used here in this data set, and then there was the whole soil fraction. And the K feldspars were only reported in the whole soil fraction. And the difference of abundances across the K feldspars was pretty minute. There wasn't a whole lot there in as far as the range and difference. I did try to model it to make a map, and it spectacularly failed. So possibly because it's just there's not enough variability, or it's not captured well enough. We did look at it, but I didn't focus on it simply because it was just hard to see differences between the feldspars.
So I'll ask another question. You mentioned something about pH, that you were doing some measurements on there. Can you just talk a little bit about why we're interested in looking at pH on some of the sandier soils?
Yeah, so we were looking at that pH, kind of assuming that as you increase your pH, that you would increase your negative sites on the soil, and therefore you'd be able to sorb a little bit more if you raise that pH in the sands. But we really didn't see a whole lot of difference. There was a slight significant difference. Well, there was a significant difference with the calcium chloride, but it seemed like what was driving that calcium chloride was actually desorption. It wasn't really capturing any sorption. And the relationships were kind of mixed, but essentially, I mean, the initial thought behind that experiment is maybe the pH and that variable charge from the pH in the sands can impact our CEC and therefore our potassium retention in sandy soils.
Did you adjust the pH, or did you just go out and find soils that had pH?
We adjusted it.
You did adjust it.
Yeah.
Okay.
So we started with the lowest pH, 4, 5,
Okay.
and then we limed up to a medium and a high. And so, part of this could be the lime may have played a big role also. Liming, has a lot of calcium carbonate in it. So you're already kind of knocking that off the site, which we, didn't anticipate to see that huge difference between those two. Yeah.
Are there any questions online? I'm not seeing anything in the chat.
I just had a question back.
Oh, no.
So with supervised regression, supervised classification, something that I often get asked about how many samples you need to fit a model. So after going through this process with clay mapping and soybean prediction, how would you answer that question? How many samples do we need to get a suitable model to take to the next step?
You might need to repeat that.
So the question was about, if we're doing these supervised regression problems, common question that people ask is, okay, well, how many samples do you need to build a model that's accurate? And that totally depends on the data. I mean, I would say, give me as much as I can possibly get my hands on. There's no end to improving your model with more and more data. So you just want to always keep collecting more. There's not necessarily a set number of this is the right amount of samples. The quality of the data and the quantity of the data are important. And, one of the important things that I came across too in doing this is that the algorithm, while it might seem important, sometimes the algorithm is less important than actually the quantity and the quality of your data. So, in chapter 3, I use LAAS, which is a simpler, more simplistic model, supervised regression model, and then used random forest, which is a little bit more complex for the soybean. But I've seen papers too that have been comparing different models, and really this is a data-driven process. So the more data you have, usually the more accurate your model can become.
Are there any other questions? Well, I think we'll wrap this up for today. So again, let's thank Leanna for her presentation.
Thank you to everybody online.